introduce Jorge Fernandes. He's from Portugal originally, and he did his bachelor and master degree there, and then went to, as a part of a PhD, to a prestigious place or college, university, which I always forget the name. It's like, sounds like Guggenheim, but it's different. It's Guggenheim. Guggenheim is a elite place in Portugal. The top students are allowed to go. And he was admitted there and did part of the PhD there. He got his degree first in chemical engineering, and then went to this prestigious school, and then went to do his PhD in St Andrews in Scotland. And after that, I don't know if you went somewhere else, but for the last 15 or so years, he has been a professor at the World University in Norway, in North, there, close to the Polar Circle, where he has built an impressive team, and where he has been awarded 2-year sea grants. And last year, during the strategic committee, so he applied he got the okay to apply for the clear position, which unfortunately we not get. And then this year there was the chance of applying for one of these like open positions as a full professor in terms of core interdisciplinary science, which was very competitive. There were like 40-something people showing up, and he ended up having securing one of the 4 positions that were relevant. So with you, Jorge, and I hope that you enjoy your talk. We will.
Thank you. Thank you very much for the introduction, Francesca. Even though now that he has actually spoiled my talk. Because the beginning of the talk is about quite a bit of it, about my CV. Thank you very much. And thank you very much to volunteer and the rest of the direction for giving me the opportunity to come here and visit the Institute this week. I really appreciate it. Also, the title of my talk is a bit misleading. This is not going to be a scientific talk at all. Well, first, there was not much time. And second, I thought it would be best for you just to know a little bit more about what I've been doing, what my competences are. And throughout, I'll give you a few examples or show you some results that we have obtained in different fields, but just give you an overview or a taste of what I've been doing. If you have any questions and you want to know more, I'm happy to talk to you afterwards. So, you can know this is not working. Now it is. Okay. So I started doing my bachelor's in engineering here at the Institute Superior Técnico in Lisbon, in Portugal. And towards the end of my degree, there was a professor of mine in thermodynamics, and she told me, why don't you go on to do a PhD? At this point, I had already been offered a position in a company called Ovion that does pharmaceutical things, and it was to go to Macau, China. I hadn't really even heard much about PhDs. I knew what it was, but I didn't really know much about what it involved. And so then I went to France, to the Center of Nuclear Studies in Saclay in Paris, to spend 3 months doing some lab work and to see if I liked it or not, and I really did. And I also started to become very interested about biology. And that's when I went on to join this Gulbenkian's program for PhD in biology medicine in '97 at the IGC, Institute of Gulbenkian Ciência. This is very special, and again, it contributed to my interdisciplinary background because they hire or have scholarships for about 15 people from the whole country in any field. So I had, for example, 2 colleagues that were physicists. I have several medical doctors. There was even one lawyer, but that one actually quit. But it's a competitive program, but it's also very good that you are interacting with people from very different backgrounds. I mean, truly interdisciplinary course. And it's also very tough because we have 2 weeks of evolutionary biology and 2 weeks of ecology and 2 weeks of this and that and the other. So we cover a lot of fields in biology. And for someone who is coming from a chemistry background, it is really tough, but also very, very exciting. And from here, I went to Scotland, to St. Andrews, to do a PhD in comparative immunology. This was mostly biochemistry. I have a couple of slides about the PhD. And then I wanted to do more in molecular biology, which was still fairly new in non-model fish species back in 2003. So I thought this was a good opportunity to learn something that will be at the forefront of the field at that point. And I studied, or I moved to a different lab in the same university to do a postdoc in muscle development. And in 2007, I moved to Buda, which is really up there. So Oslo would be around here, Trondheim here. Where I have been, well, when I moved there, I was an associate professor in molecular biology, and then in 2010, I got promoted to a full professor in genomics and bioinformatics on the basis of consorts. There are some of these self-promoting things because this was for the opposition. So sorry, I tried to remove most of them, but sorry, Austin. Okay. And I put a picture here of my PhD supervisor because she passed away last year, unfortunately. She was really nice. Yeah, now over a lot. Was Dr. Valerie Smith. She's a comparative immunologist. She was actually an expert in invertebrate immunity. Mine was her first PhD or first project in invertebrate. Yeah, I think we did quite well. And the aim of my thesis was to look for antimicrobial peptides in mucosal surfaces of rainbow trout, especially on the skin. And these antimicrobial peptides are very short peptides or proteins, up to 10 kilodaltons in length, just a string of amino acids. And they have a number of very interesting properties. They can be antibacterial, antiviral, they can stop tumors from growing. They have a lot of different properties. It's amazing that such a small thing, sometimes they are just 10 amino acids long, they can have this kind of effects. And you're doing this in rainbow trout because, as you probably know, it is one of the most important commercial species in Europe. And because the innate immune system in fish is very, very important, you can, or you may have read that fish don't have a very sophisticated immune system. I disagree. And even the adaptive immune system in fish is actually quite good. That's why they've been around for so long. But nevertheless, the innate immune system is very, very important. And these antimicrobial peptides, because they are so small, they can be made very quickly. And they're normally found on the mucosal surfaces like the skin, the gills, the gut, where the fish actually, or it's the first line of defense against invading pathogens. So it's the places that contact directly the microbes in the water. And yes, so these peptides are very, very important for the innate immune system, also for us, and also because they have potential applications as natural antibiotics. So far, or as far as I know, they are not being used commercially in aquaculture, but there have been several trials, clinical trials in humans, for example, to use these peptides to treat diabetic ulcers and other serious infections that can be treated topically. Okay. This was just a little bit about my PhD, again, to emphasize this multidisciplinary approach, because this position is in interdisciplinary science. So basically, we collected the mucus from the fish. We did several fractionation methods, like different types of chromatography. And then we ended up with a lot of fractions that we needed to test using antibacterial assays, this microbiology. And then we would pick these ones where you see these clear zones the bacteria are not growing. So they are being killed, or at least not growing. I would run protein gels of these fractions, and then do another chromatography and so on until I ended up with roughly one antimicrobial peptide. And then we would try to characterize it doing protein sequencing. In those days, it was very, very, very tricky. You get maybe 10 amino acids or so, and we also use the mass spectrometer to look for protein modifications and so on. And that's an example of functional assay where we incubated this peptide with E. coli. And as you can see here, the bacteria is completely dissolved. If I show you a control picture, it would be just like a normal rod. Okay, so sorry, I don't have this present for you, so I have to do this. Okay. And one of the highlights of my PhD is that I was the first person to show that histones, especially this histone H2A, are very strong antimicrobials. So most of us will know that histones participate in the packaging of the DNA inside the nucleus. But these papers that I published actually show that histones are very important for the innate immune system in rainbow trout, and not just there have been many other publications after this. And actually, it's funny that I had this old British co-supervisor that was doing the sequencing for me, and I would send him the instructions, and he would sequence them. And when he got this result, he told me, "I've got good news and bad news." And okay, good news first. The good news was that finally managed to get a very good sequence from what I gave him, like 7, 8 amino acids was pretty good in those days. And then, okay, so what's the bad news? The bad news is that it's not what we are looking for, it's histone. So he was very surprised and he thought it was just a... Pardon?
Is what?
Histone. Histone. So it's these proteins that are around the DNA in the nucleus. And he just dismissed it, and I was just very disappointed. But then I did it again, and we got the same thing. And again, and the same thing. And then after this, I found another histone like H6, for example, that got the same result. So then he was convinced, and I was also convinced that these histones are antimicrobials as well. And some of them are only produced, or these fragments, after the fish is injured somehow. So we injure the fish a little bit with sandpaper. It's not as bad as it sounds because it's really just a little bit, so it was not bleeding or anything. And then the expression of these histones increases a lot, or the production of these fragments. So the protein is there, but it is cut to produce these small fragments. Okay, and these are just some details of this histone that is similar to other antimicrobial peptides. It is sensitive to salt. It does not kill the red blood cells of the trout. And in this case, it does not form stable pores on the membrane because some peptides actually, so if you imagine the cell like this, they will just insert themselves in the cell and they form these holes so that the contents of the cell actually just leaks out. And other things like salt, water, whatever, can actually come in and disrupt cell physiology and they just die. But these ones do not form stable pores, so they must be going in the cell and just interacting with some cell machinery involved in replication or translation or anything else that will make the cell basically just die after a while. Okay, and then I don't know what's going on here. And then okay, maybe we can skip this part. And then for my postdoc, I was looking for genes that are involved in muscle fiber recruitment in the Japanese pufferfish, this technical varieties. I was working with Ian Johnson, who is a renowned expert in fish muscle. And you probably have heard about this fish, maybe not the full name, but this Japanese pufferfish that is poisonous and that they like to eat it in Japan. It's actually the poison is found only in the wild fish because of the algae that they eat, but they also have started to farm them. They are not as exciting as having. It's not as exciting as having wild fish, but it's still pretty popular to farm food when it's very, very expensive. Okay, but maybe yes. So I was doing some. At the time, cutting-edge molecular techniques like this suppression subtractive hybridization, where we make these libraries, we clone all these cells, we plate them, and then we pick by hand hundreds of bacterial colonies, and we make mini preps and Sanger sequencing and so on. And to get about 100 sequences, it would take maybe 1 month. But at the time, it was really, really high-tech what I was doing. I also, at this point, I learned by myself bioinformatics, and after a while, I was administrator of our unit server. But the main thing is that I got really fascinated by fish muscle. Because the fish muscle is very similar to mammalian muscle, but in many ways, but it has many particularities that make it very, very exciting thing to study. So, there are two types of muscle, two types of skeletal muscle. One is called red muscle and another one is white muscle. So when you slice a fish, most of it, let's say if you slice a sea bass, most of it will be white, but on the sides, here on the lateral line and on where the fins are, you'll see some darker muscle that we call red muscle. And this separation between the white muscle and the red muscle does not happen in mammals. So our muscles are all very mixed. You tend to have both red and white muscle mixed together, except in facial places. So if you want to understand how the fast muscle works or how just the red muscle works, the fish are a very, very good model for this. But what I find most interesting is that many fish species have indeterminate growth, meaning that they continue to grow throughout their lives by increasing the number of muscle fibers, not just the size of the fibers, but also the number of fibers. Whereas we, like all of us and the baby, we have pretty much the same number of muscle fibers. The only difference is that we have been growing our fibers since we were born, and but the number itself is pretty much the same. Whereas with fish, they have the ability to increase the number of fibers continuously. And they have to do this, if you think about it, because this fish can reach 4, 5, 6 kilos. And when they hatch, they are just a few milligrams. So we were talking about a difference in weight of several 100,000 times. So it is not possible to do this just by increasing the number of fibers, because it would get to a point where you cannot get oxygen and nutrients that you need to make these fibers work. So you need to make more fibers. This, of course, will have a lot of applications in human clinical medicine. For example, people have strokes or other muscle injuries. But I just find it interesting to find out why, or I was trying to find out why this happens in fish, but not in mammals. So by using this technique, we could compare fish that are still recruiting fibers very actively, like this small one, this not that small for the juvenile, and then a very old fish that has reached sort of a plateau in the number of muscle fibers, and finding out which genes are being differentially expressed. And we found 4 genes that were associated with this increase in the number of muscle fibers in the Japanese pufferfish. Of course, we didn't do any functional work, so I don't know if they were really doing this, but there was a link between the expression of these genes and the growth status of the fish. In particular, we found a new gene that is called C, conserved edge express protein. And this gene I found really interesting because it is present in pretty much every organism that we look at that have its genome sequence at the time. We don't know what it is. And every now and then I remember I still go to Ensemble or other databases of genomes and try to find out if anything else has been done on C. It's still not much, but it is expressed in particular places in the muscle. So again, if we think about the field of the myotome, of the bulk of the muscle, so it must have some specific function there. It's not even ubiquitously expressed, and it's not expressed at all stages of development either. So it must have a function. We don't know still what. And I found also several splice variants of important transcription factors involved in muscle growth that were changing with temperature and affected by temperature, especially the temperature at which we incubate the eggs. Okay, this was just to say that I had this honorary professorship at the University of Tokyo in 2006, and this was a very big honor because most professors in Japan, by the time they are promoting to professors, they are almost retiring. It was very strange. We had these professor meetings every month, and I was the only one that had less than 60 or so. But yes, and there was a point in putting this slide here, but I cannot So I have been at Noor University since 2007 as an independent PI, and I've been working mostly on aquaculture, on various aspects of agriculture, but especially in immunity from my PhD background in growth from this postdoc, and then in epigenetics since about 2010-11. Because I wanted to see what is the effect of the environment on growth and immunity through, or what are the epigenetic mechanisms involved in this effect that we can see. I don't know how much you know about aquaculture, so I don't know if I should skip this or no. Okay, so I have 2 or 3 slides in. We also try to advertise aquaculture, and it is true, as one of the fastest growing food sectors in the world. And it is actually poised to increase because the number of people just keeps increasing exponentially. We need good quality protein, and aquaculture is a very good alternative to provide this. And it's not as bad as meat. Nevertheless, there are reports stating that a third of the available commercialized species are overfished and captured unsustainably. And that's another reason. If you look at this graph of production along the last 6 decades or so, you can see that there has been a huge increase in aquaculture production from the 80s, like exponentially, whereas both marine capture and mariculture and inland capture, they have been reaching a plateau or increasing trade. So we actually, since we expect the world fish consumption to increase by 18 to 20% by 2030, we need to also increase aquaculture production. But to do this, we need research, basic research, and technological advances as well. It is amazing, and I didn't even know this myself until June when I had prepared for this, that before 1990, there were hardly any papers on aquaculture research. So especially in marine aquaculture, very, very little. But it has been increasing exponentially since then as well. See, the numbers are still very, very low. This is the number of new publications per year. And if you go back to the 90s and early 90s or a little bit before, most of these papers, they are just individual contributions from single disciplines. People will be looking at their nutrition or reproduction or ecology or health or economics. But now, we know, and actually there are several white papers on this, including from the World Aquaculture Society, but also from the EU, that sustainable growth of the industry, of the aquaculture industry, requires a holistic approach across different disciplines. Because the problem is very complex, and I will show you a slide where it's very clear why we need to know so much about so many different subjects, or try to integrate so many different subjects. And so, like Engel stated in 2016 in this white paper from the Journal of Aquaculture Society, that we need interdisciplinary research to improve the competitiveness, production efficiency, economic viability, and long-term sustainability of the aquaculture industry. It's a very simple diagram of production chain, where we start, of course, with wild fish. We have to somehow develop the process to produce them in captivity, and then we have to bring them to the table to a lot of transport, marketing, and stuff. And these are just some of the disciplines, and this is not extensive or exhaustive tool that are involved in this process. So maybe I'll just go back, sorry. So we have aspects of evolution, ecology that need to be understood, and physiology that need to be understood for that particular species. We need to understand how they're reproducing wild and how we'll manage to get them to reproduce in captivity. Of course, immunology is very, very important because the fish in aquaculture conditions, they are too many fish in very small space, very prone to diseases, some diseases that they don't even catch in the wild. So we need to develop vaccines and so on. Of course, there are many aspects of engineering, from the tanks to the feed and so on. Nutrition, so we need to provide the fish in captivity meet the correct nutritional requirements, similar to what they would find in the wild, similar as possible. Of course, microbiology as well, genetics and epigenetics, and then more from this side, we have food science and toxicology, and social science and economics. And there are other things that are missing here. And I've actually been working in most of these aspects, except engineering and social science and economics. I've actually put my finger in all these other types that we can see. And I've done this in 23 research projects. And two of the main projects that I had were these two projects that was called ProCode, financed by the Research Council. I got this just 1 year after I moved to Norway. It was this Young Investigator Award. And what we wanted to see was to determine or understand the molecular networks that regulate sexual maturation and growth in Atlantic cod. So Atlantic cod used to be one of the main farm species in Norway. And Norway was the largest producer of Atlantic cod worldwide. The industry actually dropped down shortly after I got this project. Hopefully it was not, well, it was not because of my results, because it happened during the project. But actually, it has started to pick up again. So the government started to invest again last year in cod aquaculture and subsidies and research. So I think that we will see the cod farming coming up. But anyway, the problem that I was addressing here has to do with the fact that the Atlantic cod reaches maturity at around 2 years of age. And they do this just a few months before they reach the market size. So we need to keep them, or they need to keep them in the sea cages for another 2-3 months, you know, feeding them, space, electricity, people. So this represents a huge cost for the agriculture industry. And because the cod fisheries was actually doing quite well at this point, these 2-3 months were enough to cause a loss. So the industry was not even breaking even. There was actually an economic loss if you wanted to, when you were farming cod. And so we thought that if we managed to find out a way of getting them to mature a little bit later, after they reach this market size, about 2 kilos, then we're saving a lot of money to the industry and actually making the industry profitable. Okay, I will not tell you any results from this, but and the second one is this, the epifish project that was funded by the European Research Council. And the idea behind this project, the research question that I wanted to address in this project was, what is the role of epigenetics in fish domestication? So it was a very simple idea, and it can actually, well, I actually had the idea first, and then I went on PubMed to check what was known about epigenetics and fish domestication. And it was like there were actually no publications whatsoever, 0. So I thought, okay, this must be new, since there are no papers about it. And that's where I started to develop from there. Basically, I guess that you all know about epigenetics here, but there are 3 main mechanisms: modifications of DNA, modifications of histones, and non-coding RNAs that can regulate or modulate gene expression in response to environmental changes. Not only, but especially to respond to environmental change. So epigenetics plays a very important role in the interaction between the genotype or the phenotype and the environment. So what we did here was to set up a small domestication program. We went to Egypt to collect wild fish, wild Nile tilapia. Sorry, I forgot to say this was Nile tilapia. The Nile tilapia or tilapias are the second most important fish produced worldwide, after carps. And also it has other advantages for this project, that the genome has been sequenced. It has very short generation times, just a few months, like 3-4 months. It was very useful to look at many generations of the fish throughout the project. And so we went to Egypt, collected wild fish, and then we brought the eggs or larvae back to Norway, where we started this small domestication program. Basically, we had 2 lines. One was with fish that had average size, and the other one where we picked the fish that were the biggest at a certain age as parents for the next generation, and so on and so on. And we could see differences in size of sometimes 10-20% in just one generation between these average size fish and these ones that have been selected. And then from here, we just compared the control fish with the selected ones in each generation, and also across generations to see what was happening to the gene expression and the non-coding RNAs, and also to the DNA modifications in this fish. And then we were hoping to find some epigenetic markers of growth that, if we validated them, they could actually be used to select for more, for fish that grow better, and therefore increasing the profitability and sustainability of the aquaculture sector. So still haven't given up on this, but it is still early days. I'll just give you two examples, very briefly, of two studies that we did during this epifish project. So one has to do with the epigenetic remodeling that happens during domestication in the first generation, when we take the fish from, when we took the fish from Egypt and we put them in Norway, or in our recirculating aquaculture system in Norway. And the other one is about the microbiome persistence that we observed in this first generation. Okay, I'm not going to go into any details here, but I will just explain what you see here on this circus plot. So each of these arcs represents a chromosome, or the equivalent of a chromosome, so in Nautilapia. So this whole thing is a Nautilapia genome, and each of these is chromosome. And here we can see the fish that are undergoing domestication, this generation that was reared in captivity. And here we can see the fish that are wild. And these bars represent the levels of, actually it is not methylation, it is another epigenetic marker called hydroxymethylation. And here we can see the levels of that hydroxymethylation in these specific positions in the genome of Nautilapia. And just by looking at it, you can see that there is a clear difference in some places. For example, here, we have much more hydroxymethylation in the wild fish than in the domesticated fish or undergoing domestication. And some of these actually fell within genes that are very interesting because they are involved in growth or immunity. Basically, we could just see that there was hydroxymethylation throughout the genome, and in some cases we have more or less in one group compared to the other. And again, this is the first time that we observed these changes in hydroxymethylation in the muscle in any fish species after one generation of domestication. And like I said, these genes involved in immunity, growth, but also neuronal related patterns. The other study was about the microbiome in the mouth of wild Nautilapia compared to the ones that we had in captivity. We decided to look at this because Nautilapia are mouth brooders. So the females, after the eggs get fertilized, they pick them up and they keep them in their mouth for ages until the hatch, or even actually some days until after the hatch. So you can see these fish, they go out to eat and then they actually even come back again when they're already quite big. Anyway, we wanted to see if there was anything special about the microbiome in the mouth of these fish, of the females, and if these bacteria were kept, even if we are breeding fish in Norway in a recirculating aquaculture system, compared to these fish that come from a pretty dirty place in the River Nile in the wild. So we just collected the mouth, well, and intestine as well, the mouth mucus, and we just compared them using this traditional 16S metabarcoding. And we could see that, just to summarize, that these three genus of bacteria were transferred from the mother to the offspring. We could find them even 2 generations after they had been reared in captivity. And also, of course, and this is a limitation of the 16S studies, we don't know exactly the strain of this bacteria that we are looking at, but these bacteria are reported to have or to play a key role in the health and development of fish. So we think that they are being kept because they might be important for the early development of the offspring. Okay, so almost there. The research that I have been doing and that I plan to continue doing has been addressing several challenges in European agriculture. I was looking at the current challenges, or what they predict will be the challenges in the next few years on this European Forest Strategy and Circular Blue Economy, published by the European Commission. And they mentioned climate change, depletion of natural resources, loss of biodiversity, food security and safety, and environmental pollution and waste. And all they suggest that we should address these challenges is by increasing resilience and adaptation to temperature, improve the selection for robust fish, change the diets that we have been using now even more and more to new plant and insect-based diets to avoid fish meal and fish oil, and to reduce the use of antibiotics. What I plan to do in the next few years is to continue more or less what I've been doing, looking at the effect of diet and temperature on health, growth, and the microbiome, but through an epigenetics perspective. So I want to focus on holoepigenomics. So there is not much on holoepigenomics at the moment, not even in hologenomics in fish. Basically, as you probably know, the whole genome is the whole sets of the genomes in a holobiome. So it will be the host genome plus all the thousands of other genomes that we have, and that are very important for our phenotype and physiology, everything, for growth, everything. And we know that they are involved in the evolution and fitness of the organisms, and we know that they're affected by the environment. So I want to see how environmental and nutritional factors affect the whole genome, but not just that, also the epigenome and the meta-epigenome. So because so far, again, there's nothing known about the holoepigenome in fish. Even the word is not very common, and I think that it will be very good for us to go beyond what we are doing now and look at epigenetic interactions between the host and the microbiome. And I will also focus not only on the traditional epigenetic mechanisms like the methylation and chromatin accessibility, but I want to look at things like hydroxymethylation and other types of non-coding RNAs, like this circular RNA that you see there. And when it comes to methods, I will move more to SMRT, like a single molecular real-time sequencing like nanopore and PacBio, including for the sequencing of bacterial genomes, and single cell or single nucleus PacSec for chromatin accessibility and RNA-seq. And I hope that soon we will have single cell DNA methylation analysis as well. Also, things like spatial transcriptomics and epigenome editing are some things that I would like to be doing in the future. And yeah, I think that is basically it. I think that this will really bring our knowledge about the epigenetic modulation of immunity and growth to another level compared to what we know now in fish. And this can actually have some applications, maybe not in the immediate future, but in the near future to the aquaculture.